Okay, so I will present on the previous paper we published last year, um, really looking at the phosphosignaling across breast cancer. And let me see how I get this into a full screen. And I'm Kuan. Everyone here probably knows me. I'm at a computational omics lab at Icahn School of Mount Sinai in New York City. And before I get into the talk, I think it's actually more important to, can everyone see my share, by the way? I think it's not, maybe it's not working. Resume share. Can everyone see my share?
I can see the front slide. Oh, now it's going, yeah.
Okay, so maybe I cannot full screen it. Okay, thanks, Tommy. So I think I previously talked about this within the lab, but I think just more people are thinking about the conceptual framework, because I think actually from Lai has talked today, I think in science, one of the most interesting thing is to realize what conceptual model you're operating at and what's the advantage of that conceptual model and what could be wrong with that model. And sometimes, most of the time, the biggest opportunities are if you want to do some paradigm breaking science, it's about trying to look at what existing paradigm models cannot find. At the same time, it's a little tricky because sometimes you first need to get some credential, and then you have to be operating within that paradigm model and be the best in that model. So a lot about how I think about how precision medicine work. So in Laha's talk today, Brian's talks earlier, like you have different kinds of big data. You have all the DNAs. In Brian's talk, you have like the 300,000 dB gap SNP, and that captures a lot of human variations at DNA level. Some of that get transcribed into RNA level. And that can also be proteins, which we'll talk a little bit more about. Or in single cell experiments, that can be different kind of cells. But this is too much information in some ways. And to have diagnostic biomarkers and therapeutic targets, since molecular biology and genetic approaches really have this kind of reductionist approach where you try to find, once you have the whole profile, now with computational approach, you can find a global patterns that say these genes are expressed more in tumors, these genes are expressed less in tumors, these variants are seen more in tumors, or they are seen more in non-cancerous people, or these variants are seen more in COVID patients that end up doing better, then maybe those genetic variants are protected through some kind of molecular mechanisms. So you can start seeing these global level patterns, but really also identify the outsized agents that have disproportional roles in driving the phenotype you're interested in. So for example, in a society, some individuals may have outsized contribution to how that society function. And that might result into eventually whether that society operates in a healthy way or not, or an organism, whether that organism develops certain disease or not. In a lot of our disease studies, if we can identify these specific events of interest, then that could be potential diagnostic biomarkers as well as possible therapeutic targets that eventually now in a lot of targeted therapy we develop is really aiming for these targets we can identify through the global omics studies. And so next time when you have the next patient come and generate this sea of data, you can really find the precision diagnostic for the patient and have targeted therapy for each person's disease. That paradigm has worked pretty well, and I would say that in some cases, at least in cancer, it works pretty okay on DNA level, where for a lot of mutations, we already have targeted drugs. So patient A may have mutation A. You then use drug A that target this mutation instead of drug B that targets mutation B that you apply for patient B for the optimized outcome. But this framework still has a lot of caveats. I think one of the most important caveats is that we really know from the central dogma, in most cases, DNA goes to RNA before that gets translated to protein, and protein is the one that carry out the majority of cellular functions. Because of this, most of our drug, whether they are small molecule compounds or antibodies, which now in this case of COVID, a lot of them are also antibodies, really target the proteins. So if the DNA completely correspond to protein, this paradigm would have no problem, because if you find a mutation, you find a drug that will work for that mutation manifested at a protein level that could work. And that has worked for things like BRAF, B600E, some of the EGFR mutations in lung cancer. But there are a lot of cases where that genomic mutation may not manifest as we expected. And also, there could be also these black box that say different disease like cancer utilized in the middle, so that now you have aberrant proteins that are not really found at the DNA RNA level. And so a lot of our current research are trying to address this problem. Maybe, at least for now, before we figure out these black boxes, we can have better targeted diagnostic and treatment from understanding the protein level better. And so I think all the talks today really covers a wide ground. Laha's talk really discussed the germline variants and the somatic mutations, which are great and important, and already some of them are implemented in healthcare, but Laha's really show us we haven't really considered, especially a lot of the germline variants that could be important. And in Brian's talk, we talk about how they are different mRNA variants that reflect these DNA level mutations, that through efficient computing and sharing, we can really then query these RNA for the variants that are really reflected at the RNA weak count level. And that can really help us validate some of these genomic variants in their gene expression impact, and potentially discover other ones that we didn't anticipate to have gene expression impact. And actually, Brian, we're actually collaborating with Hannah to use some of these data. So thanks a lot for that. And then finally, I think I will discuss a little bit about translating this into protein, and especially to the signaling network. And this really is empowered by the new mass spec data we have. I will not really get to the data part, but I want to start a little bit by motivating on a biological part. So in terms of biology, we know that there's a lot of signaling pathways in cancer. And these are well identified, characterized by a lot of functional studies to have functional effects. And so you have Wnt signaling that may go wrong, you have RAS signaling that may go wrong, you have autophagy might go wrong, you have AKT, PI3 kinase signaling that might go wrong in cancer, right? So there's a lot of possibility. The question is, when we see mutations, sometimes we only see them in specific pathways. So does that mean that, in all the cancer, do all of them go wrong? Probably we don't think so. Probably we think that in some cancer, AKT signaling is upregulated, and that contributes to cellular overproliferation in survival. In circumstances, they should not survive. In some other cancers, they might have RAS just really upregulated. So they're all having different signaling pathway being light up. If you think about how it might look, one cancer might have this signaling pathway light up, and the other might have this one light up. Previously, we kind of rely on genomic to infer this. If we see, let's say, a PI3 kinase mutation, we would try to put inhibitor that inhibit this pathway, and hoping that will work. For the case of PI3 kinase hotspot mutation, it's quite interesting because in functional study, that mutations are really validated in vivo and vitro model, but actually the drug doesn't work that well for PI3 kinase mutated cancer. And that could be really potentially be attributed by the black box that happened between DNA mutation and signaling. So really the goal is now we have these rich mass spec data of 10,000 proteins, 30,000 phosphosites. Can we start identifying how these events are actually correlated in patient's primary tumor, and how that signaling actually works, instead of based on the prior knowledge of how that signaling have worked in vivo and in vitro models. And so this really allows us to ask the question, what signaling events are really upregulated in each primary patient's tumor? And what we used the data set here was the Mertens et al. 2016 Nature data set, where they published around 105 breast cancers that had mass spec data, 77 passed QC. Among those, around 30,000 are quantified in more than 10 samples. So if you think about the scale of the data is really interesting because before this, pretty much we use ELISA or RPPA, where you survey like a couple hundred phosphosites at max to see their quantification. But now we suddenly see 30,000 of them. So how do you prioritize them? And how do you know which ones are really turned on? And now this experiment is so rich that in this data set, for example, if you compare to what phosphosites are available on phospho-ELN, which is one of the major database, and UniProt, which is another major database, there's a lot of them that are novelly identified in just this mass spec run. And now we actually have this for 10 cancer types. And so the idea we had was, this data is really huge to begin with, but we know that in signaling, the kinase and substrates are really important. The kinase are known biologically to phosphorylate the substrates, and the cascade of kinase substrate to the downstream substrate really turns on the whole signaling pathway. Using this data set, there is a caveat though, because the mass spec quantification works by comparing within the cohort the relative abundance. So we only have the relative abundance within the cohort, which means that we only have the power to identify this kind of, we call regulated kinase substrate pair, where in one patient's tumor, the kinase is high, the substrate is high. In the other, the kinase is low and the substrate is low. We can actually get a phosphoprotein of the kinase and the phosphosite resolution at a substrate. But then that helps us distinguish from the uncorrelated one. But then we also cannot identify the one that are, say, high in all patients' tumor or low in all patients' tumor. In this case, I think we're a little bit lucky because in breast cancer, it's a disease known to have molecular heterogeneity across patients. So we kind of assume that, maybe we'll see some that are more prevalent in basal subtype breast tumors and some that are more upregulated in luminal subtype breast tumors. So it really allows to see these kind of events. So the goal is really to use a model to find these kind of correlations. This was published last year in MCP. Sorry about the slide lacking updates. So together with Iga, who is now back then a rotation student and now a PhD student in Lee's lab, we look into how the phosphorylation work with regards to both autophosphorylation, which means the kinase phosphorylate the substrates, subphosphosite upon itself, or transphosphorylation, which is more what you expect in more cases where kinase phosphorylate a substrate phosphosite on a different protein. We kind of loosely identify cis regulation because we're not using the strict role of autophosphorylation where we validated this is actually the site of phosphorylate itself. We kind of just see whether the phosphoprotein level of the kinase overall correlated with this specific phosphosite level. And so here are 3 phosphosites, for example, along the AKT1 protein, which is very important in breast cancer. It's one of the major kinase and substrate in the PI3 kinase signaling cascade. And it has drug compounds developed for AKT inhibitor, and its upstream PI3 kinase inhibitor, and its downstream mTOR inhibitor. Within even just this protein, you can see that the substrate phosphosites, the correlation actually varies. And these 2 sites, the threonine phosphosites, are actually more surveyed by current antibodies because we have good antibodies for it. So we have been relying on it as a good gauge for AKT phosphoactivity. But as you can see, it actually does not correlate with AKT phosphoprotein level that well. And we think in this case, at least, S129, which is serine 129, is probably actually a better phosphosite surrogate for this kinase protein activity potentially. And the activation is a little subtype specific, but not that subtype specific to luminal breast tumor, which are known to have more PI3 kinase mutations. And that potentially maybe also explain why I think since 2018, the earliest trials in PI3 kinase inhibitor and mTOR inhibitor actually shows that PI3 kinase inhibitor does not necessarily make PI3 kinase mutated tumor having better outcome. It's not correlated with the mutation at all, which is kind of interesting observation. And at the same time, besides from the well-known autophosphorylation protein kinases, we also find some of the more new signaling event that we kind of suspect that happens between different proteins that previously might just have predictions. So before this way of using quantitative data to link kinase to substrate, one of the previous way to link, or I guess the previous most prevalent way to link computationally was based on kinase binding motif prediction, whether you just based on the substrate sequence, predict whether your kinase will go to that substrate site and phosphorylate that site. But now we really have quantitative data to really back this up more. And even the one we find in quantitative data actually show an enrichment overlap with those people previously predicted. But now we have evidence that this is probably what breast cancer is actually using to link the kinase and substrate. So for example, CDK1 and lots of CDKs, we see they activate some of the downstream substrates at these specific sites that correlated pretty well. And then lastly, I think one of the important insight we have was that we kind of wanted to try building out the signaling cascade, because previously we're really functioning in a single driver paradigm where if you see an EGFR mutation, for example, you just try to drug that EGFR. But now if we have phosphosite data, we can check its downstream signaling to see that, okay, maybe in this sample, I won't discuss the heat diffusion mechanism algorithm, but it's widely used in network biology to link multiple level of correlations, that we see that, for example, EGFR is tied to these specific downstream phosphosites, PTK2, FAN129, but PTK2 phosphorylation is also further tied to these downstream phosphosites. And so does this mean that maybe in the future, when we identify a key driver mutation or upregulated protein, it might actually be worthwhile to check whether the downstream phosphosites and phosphoproteins are also upregulated? Because normally we just drug and inhibit this EGFR, but maybe that tumor used the EGFR early on in cancer development, and now it's relying on some other mechanism. So once you inhibit it, it actually doesn't care because these downstream are like already tuned off. But if the tumor is still actively using it, you should see this in all red. It should all be upregulated. Then in that tumor, if you inhibit that EGFR, you are more confident that you are inhibiting this downstream cascade, and that might have more correlation with the downstream tumor phenotype. And so this EGFR example is one example that we see the activation is higher in basal subtype breast tumor, which now have a relatively few treatment options. So we kind of suggested maybe EGFR inhibition could be a way to think about triple negative breast cancer, et cetera. But obviously, this is a very basic science research that we conducted here. So I think that's the overall of the 2019 study we published. And I think a lot of the cancer genomics and computational biology, as we see in all the talks today, really ties in all the way from using all the possible molecular portrait information. At least now within cancer cell, we still have a lot to discuss outside the cancer cell that really guide us into precision diagnostic and treatment, whether that's mutation at DNA level, mutation found at RNA level, expression found at RNA level, protein and phosphoprotein to signaling pathway. I feel eventually all these need to be jointly considered in finding the right targeted diagnostic and treatment. So I think that's it for the talk today. Some of you guys probably hear me talk about this, but please feel free to spread the news, because now we really just started this Open Box Science website, and you guys are the early adopters that I think eventually would be great. You know, like if you're gonna spend 30 minutes reading a paper, might as well just invite the first author to give a talk, and it's more easily absorbed. You can ask them questions, keeping it interactive, and people can practice talking too. I think it's a very important skill for scientists nowadays, especially, you know, nowadays I feel it's more about not just generating the knowledge, but also communicating the information. Because as you can see, anyone can say anything on Twitter, and eventually the information that wins might be the one that communicated to a wider audience than the right one. So as scientists, we have responsibilities to communicate it better. And now the upside with this crisis is now everyone's really, as you guys are, like very comfortable with Zoom conferences. So there's a lot of great talks online already, and we try to organize as much of them as possible. But if you guys want to organize them, please feel free to do so and let us know. We can help broadcast. I think we also need to work hard to try to increase the reach of a platform. So if you guys can help or think of other ideas, please let us know. But you can listen in, find the schedules. It's on Google Calendar. You can sync it with the Google Calendar. Works great. And then also, if you have a paper that you think is important, published by some other people, we actually write an email template that you can use to like invite people to present and use the name of open box science, and they can just feel more, you know, instead of just talking to you, like presenting to potentially an audience. And, you know, nowadays it's pretty easy to host a free Google Meet or a Zoom meeting, whatever works for you. And I think, you know, we'll try to do this at least definitely within my group in Sinai, but I think also for anyone, I think it will be definitely very important to present your published findings, practice presenting, and also promote what you find. And maybe sometimes you find like potential collaborators or questions you never think about. So I think those can all be pretty great. And so that's it. And here's a bunch of open box science information, and welcome any questions. Thanks to a lot of people here actually in the lab and everyone for listening, and feel free to ask any questions on either open box science or some part of a research we discussed, whether in the talk just now or like in any talks today. Thanks.
I have one question.
Yeah.
So you think about AlphaFold in general. So I think Google Brain, or I forgot, one of the deep learning team in Google published a paper earlier this year on AlphaFold, and they said they beat everyone again for the protein structure prediction. Do you think that will change the landscape in terms of how we generate protein data in the future? Because right now we generating like kinase data is expensive, right? My understanding is that the antibody require a lot of optimization. It's not cheap. It's easy to screw up, and then those $1,000 worth of antibodies gone.
Yeah, I think that's a great point. Yeah, I think definitely it will be game changing. So basically, AlphaFold, Brian was talking about like this. AlphaFold now can assemble protein structure out of the sequence. And previously, once people identify a protein that they think should be targeted, someone need to use either X-ray crystallography or now more prevalent cryo-EM to actually make the structure. But a lot of proteins, we still can solve the structure. But then before you solve the structure, you don't know what your compound should look like to dock. So that's like a little downstream of after. So omics, what we do is more like identifying the right protein. But once you identify right protein, people need to know its structure. So that's a little downstream of what we do. I think it has a potential to transform the downstream. And I'm not an expert in that field. Based on what I read in that paper, I think they also discussed that. Like, I think the AlphaFold resolution is around 10 microns, which is not good enough for some of the drug design yet. The best cryo-EM data right now are around 1 micron in my memory. So like, if you can get it, like that's still better. But if you can't, I think probably it's gonna facilitate them. Yeah. And I mean, it will only improve, right? So probably like, yeah, maybe people won't need to actually get a crystal anymore. Yeah, but as you can see, like Nature publishes a crystal every month, right? Like it's super. People are publishing all the crystal of basically all the COVID proteins now, because that's like very close to the cure.
Yeah.
Yeah. But yeah, that's a good note. Yeah, but I think, with quantitative proteomic data we have, hopefully we can find out protein first. I think especially in human disease, among the 20,000 protein and different isoforms of the protein, that's still a big need. In the case of COVID, like it only encodes so many proteins, so people are basically trying to see like every of its protein. So I guess it's a different question there.
The bulk of the genome are not even coding. Those are just peptides. I think it's only like the last 5,000 base pairs in the COVID-19 genome. and code the couple genes.
Yeah.
And
Yeah. Yeah, so that there's still like some stuff to be figured out there, but we actually try to do some. Tommy is doing a lot of COVID research, but we are mostly mining patient data. Yeah, but there is a lot of drug. It's exciting. I think that research is advancing very fast because the pharma and the research labs are so open, and maybe we can just tackle every disease like this, you know.
yeah.
It's like cancer and all these other diseases are killing more people than COVID every day too. There's a different sort of urgency.
But COVID is keeping us indoor, you know.
Yeah.
Like a problem in some way.
without the party and stuff,
Yeah.
you actually need. Yeah, it's true. Yeah. Anyone have any other thoughts, comments? Any thoughts, Ghost? Okay, I think if not, we can end this. And